Síguenos para más información relacionada con hormigas y no olvides suscribirte. Bueno, buenos días a todos. Buen día, buen día, buen día. Bienvenidos a el segundo día del segundo simposio Iberoamericano de Mirmecología. Mi nombre es Diana Urcuqui y los voy a estar acompañando en esta primera parte del simposio. Here we have an incredible keynote speaker. She is the Professor Patrizia de Tor. She has a PhD in animal behavior at the University of Parma under the Professor Francesco Lemoli, from whom she acquired a passion for social insects. Later, she became as an associate professor at the University of Copenhagen, and currently she's full professor at the Laboratory of Experimental and Comparative Ethology at the Université Sorbonne Paris Nord, where nowadays she's interested in cognitive ethology, in particular, learning and memory in social insects. We are so excited for your keynote talk, and I know that we are learning a lot. So welcome to the symposium, and you can start.
Thank you very much for this nice introduction, and thank you for inviting me. I mean, it's so exciting to be here. So on the other side of the country for many. I'll just share my screen. And I've been looking at your program, and it's also very exciting. Unfortunately, I don't speak Spanish so well to be able to follow, but I'll do my best with English at the moment. So as you said, and I'm now became interested in cognitive abilities in social insects, and in particular in ants. And most of my career was interested in communication. Communication is in part of cognition, of course, and I studied chemical communication for a long time, which I'm still doing, and especially in collaboration with other researchers. But since a couple of years, knowing more about what ants can do or cannot do, and the realm of cognitive ability has been very interesting to me. And so with very talented students and nice collaborators, I could do a couple of things that I will talk to you today. So when we talk about cognition, it is clear that the first things that come to the mind is, vertebrates, because of course primates that are very similar to us, they have very similar cognitive abilities, and also birds, especially corvids, that can do amazing things. But if we think twice, we should realize that brain size is not a limit for cognitive ability. So it's not because that they have large brains, the vertebrates in general, that they can do things, and many brains cannot. Indeed, cognitive abilities in social insects are well known, and they've been studied at the colony level and at the individual level. I'm interested more at the individual level. And when we look at the literature, what has been done is mostly concerning bees, the bumblebees and the honeybees. For example, bees show very complex learning abilities. The honeybees, they can solve the principle of sameness and differences, so they can be trained to hope you see my mouse. They can be trained to, recognize what is similar and what is different for example, between two colors, so in different patterns. They can integrate different concepts, the above and below, the same and the different. And they have numerical abilities, and that have been shown in many different experiments, and they including the understanding of the concept of zero. There's something more recent has been shown. And the bumblebees, they are amazing, and they also show very complex learning abilities. For example, cross-modal recognition, so that they can kind of find out that, for example, here, this kind of geometric ball is predicting the occurrence of a reward, and they learn this in the dark, but then they can recognize it in the light. And so this is the cross-modal recognition. They have been shown to be able to learn string pulling, and they can do football playing. I'm sure you know this works because they have been, popularized by the media all over the world. And more recently, it has been shown that they can solve a puzzle box, and they do this with social learning. So this is really very well advanced, complex cognitive abilities, and they still have a mini brain. So what about the ants? Much less is known about the cognitive ability of ants at the individual level. And so I asked myself the question, is this because ants are less intelligent than bees, or simply because ant researchers are less intelligent than bee researchers? Now, for example, I'm very much less intelligent than Martin Dufra or La Chica. And so maybe this is the reason why, so we should make an effort to look at the cognitive abilities of ants. And there are very intelligent researchers working on ants as well. For example, Tommy is a friend of mine and recently wrote this review about advanced cognition in ants. And among very other interesting things, he's saying that indeed we cannot really compare the ants and the bees because the cognitive ability of ants do not line up with those of bees, because ants typically use chemosensation and touch, and these are different for the visual tasks that they use when we study cognition in bees. Or the examples that we have seen that's mostly based on visual cues. So probably we need to be able to ask the right question to study the cognitive ability of individual ants. And if we ask the right questions, then we realize that ants as well as are able of advanced cognition. And for example, they can show cognitive control. So what is cognitive control? Is the repression of a response that normally is instinctive and preferred, so what the ants normally would do, in favor or a response that is learned but is more appropriate when there is a conflict between these two. And so they showed us, for example, in the very simple and common ant Lytus niger, these ants, they follow a pheromone trail, but when there is a conflict between this pheromone trail laid by nestmates and their own memory, they make the decision based on the information about food quality that they obtain by contacting returning foragers. So this is an example of advanced cognition. So what did we do in our lab? We studied quantitative cognition, tool use, learning and memory in general, and all these things interlinked with personality. And I'll show you later why it's interesting to look at the connection between cognition and personality. And to do this, we use mostly 3 species of ants, Camponotus aetiops here, Hormigas seninis, because they show tool use, and Formica fusca, because they are really nice and very common and easy to keep in the lab. So I will give you some examples. So these things starting with quantitative cognition. And so with carpenter ants, Camponotus aetiops, we studied 2 dimensions of quantitative cognition: the spontaneous quantitative judgment. So when we just look at 2 different piles of things, which is the biggest and which is the smallest, and the use of ordinal information, that is sequential ordinal information. And to study this, we use 2 ecologically relevant tasks. So the first, for the quantitative judgment, is the brood pile preference, and then for the ordinal information is landmark guided foraging. So let's start with the brood pile preference. So the question is if there is if there are two piles of brood, I mean, for example, cocoons, one with 2 cocoons and the other one with 4 cocoons, which one would be more interesting to the ants? And for this, we use, of course, nurses that normally do this behavior inside the nest. But we wanted to tell apart the different cues that the ants might use to make the difference in case they are able to make the difference. And so we used this kind of apparatus with some dummy cocoons. So these are representing the cocoons. I know they are not very, very similar to cocoons, but they are sort of little pieces of filter paper, but they were perfumed with chemical extract of real cocoons. So they were smelling cocoons. And so we tried different combinations. Here you have a combination with 2 versus 6, but we tried 1 versus 2, which is supposed to be very difficult. 1 versus 3, 1 versus 4, then 2 versus 4, 2 versus 6, and 2 versus 8. That's supposed to be kind of easy to solve. And so we had the ant in this arena, and then removed this kind of little tube that was just to acclimatize the ant for about 1 minute, and just observed how long the ant would interact with this pile and with this pile. And so we can represent the data like is represented here. So this is, for example, 1 versus 2 cocoons, and you see that the ants, they spend more time with the pile with 2 cocoons. This is the results for 2 versus 4 cocoons, and again, so in all the combinations, we found that actually individual ants were able to make this difference, and like the preference longer with the pile for the cocoons. So the question was, how do they do this? If you remember, these cocoons are perfumed with real chemical extracts of the cocoons of the colony. So do they use these chemical cues, or do they use tactile cues, visual cues? And so to try to answer this question, we then asked the ants this other question here. So for example, when we have 1 cocoon that is not perfumed, so there is no extract of cocoon on it, versus just 1 cocoon, but this cocoon is perfumed with the extract of the cocoon from their own colony, so the ants, they spend more time with this perfumed cocoon. So it is sure that they use chemical cues. But then here we have the case in which we have 1 cocoon versus 2 cocoons, but a different concentration of the extract, so that the total amount is the same. So here we have 20 microliters, and here we have 10 and 10, so 20 in total. And in this case, they prefer the 2 cocoons. So in this case, the amount of chemical extract is the same, but here there are 2 cocoons, and here there is 1, so they must use other cues also in addition to the chemical cues. And this will be interesting to study more in detail in the future. For the moment, we can say that ants spontaneously discriminate the larger 2 quantities of cocoons, and they use chemical cues, but also visual and tactile cues. Okay, so then we go to the second task. So this is the ordinal task. It looks a little bit complicated when you see it like this, but I'll try to explain it. So here we have this tunnel, and in this tunnel we have 5 different landmarks on the soil. And here there is the ant in this little tube. In this, of course, in this task we use foragers. We didn't use nurses like in the previous task. And so the ant is here for about 1 minute of acclimatization, and then we remove this tube, and the ant is free to explore. And so this sequence of landmarks changes its absolute position during 8 training trials. So see that the position is always different, but the relative position is the same. So these are the 8 training trials, and what is depicted here is when the ants are trained to landmark 3. So they find the reward, which is a drop of sugar solution, after the third landmark. So here, 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 you see that the position is always different. So the ant cannot use a position-based rule to find a reward in the test, because after these 8 training trials, there is a test in which the landmarks are placed in a completely different position. And so we record this test, and we check how long the ant, how much time the ant is spending in searching for the reward around each landmark. And so, for example, if the ant has learned that it is the third landmark, then we expect that the ant will spend more time searching here. Hope is clear. The honeybees are able to do this, of course. This has been done in the field and also in the lab. But the ants, we didn't know. So we train individual ants to each of these landmarks, 1, 2, 3, 4, 5 different group of ants. And we first looked at the kind of acquisition of this behavior. So as you see, this is the time to find the reward when the landmark is at the position 1, 2, 3, 4, 5. And these are the different groups of ants. So you see that the first time, and there are the 8 training trials, like here. So you see that the first time it takes a long time to find it, but then they need less and less time. So this is an indication that they have learned. But now to prove this, we need to test these ants with the new combination of these landmarks, and then look at how long the ant is searching for the reward that is not present in this test phase. And these are the results. So here you have for the results for the first landmark training, second, third, fourth, and fifth. And here this tube that I showed you during the previous slide has been divided in 50 different areas. And then we were just checking the time that the ant would spend, for example, around the first reward. One is trained to this first reward compared to the other landmarks, the first landmark, sorry, compared to the others, the second compared to the others, and the third compared to the others, and so on. So the red dot here is indicating the position where the reward is expected to be, but is not there during the test. And so, as you can see, well, it works quite well. So the ants are spending more time when they are trained to the first landmark to search for the reward of the first, when they're trained to the second to search for the reward of the second, the third, and the fourth, but then they kind of fail when it is at the fifth. This is not surprising, because it has been shown also in bees and other animals that there is this sort of natural limit at 4 when we are going to count. So the performance is decreased when the ants are trained at landmark 5. So the ants can count the number of landmarks that they encounter on the way to the food source. This might be useful in nature. We don't know yet. These are laboratory experiments, and we need more experiments to show that this is real counting and not, as they say, proto-counting. But anyway, it was a good starting point, because this kind of experiments have not been done with ants, but they have been done with bees. And now I got to the second point, so the tool use. This is really fascinating behavior that, of course, we didn't discover, as discovered a long time ago. And Hormigas and a few other ant species, they do this because they cannot expand their crop. And so they cannot take liquid food and bring it back to the nest. So in nature, what they do when they find this liquid food and they want to bring it back to the nest and share it with the nest mates, they drop some material, whatever they find around, the sun grains, soil particles, leaf fragments in this liquid food, that soak this material and then they bring it back to the nest. In the laboratory, they can do funny things. I mean, we can give them things that they usually don't find in nature. And so here you have a mixture of different tools, some more natural ones and artificial ones. like these pieces of sponges that the ants are happy to use. So we had a question, this was more at the colony level, like, okay, is this behavior really hardwired, or is this somehow flexible? So the ants, yeah, are a little bit flexible in the kind of tools that they choose. And to answer this question, first, sorry, I put a little bit of video to show you. So this is the bait made with diluted honey. And these are the tools that we offer them in the lab. One kind of tools is little pieces of sponges. And I hope to see the videos that you see how they do. That's just taking this little piece and ready bring it into the liquid food to let it soak. And then this is another one up. And what we do is that we have them individually marked so that we can follow their behavior. So the first question we asked was about this, is this behavior hardwired or is it flexible? So we give ant colonies, so the entire colony, choice between different kinds of potential tools, something that they never encountered. So they have, for example, twigs, sponges, like you have seen, the little pieces of sponges, strings, parafilm, piece of paper, artificial foam that's very hard, and so on. And we wanted to know whether they would, choose the ones that are more suitable to be used as tools. And indeed, we measured their soaking properties, how much they can imbibe. And for example, it is clear that paper and sponges, they are much better than parafilm. So for each colony, we did 10 trials. And here you see the different kind of materials, so the paper, the sponges, and so on, the parafilm, and the number of these items, which different material that were put into the liquid food at the first, the 5th, and the 10th trial. As you see, there is an increase in some, even in the parafilm, but particularly for the paper and the sponges, they do increase a lot. I mean, because these are good tools. So indeed, they show preference for certain material. They are able to find out which tools, even if they are totally unfamiliar to them, like this piece of paper and sponges that they encounter for the first time. And the familiarity with this material plays a role because they use it more and more as the time goes, they can become familiar with that. And there is this preference for these artificial tools with good soaking capacity, as here the paper and the sponges. So they can be selective, they can be plastic. So this behavior is not at all hardwired. It's not like they answer little robot that do this. But they can learn and improve this behavior and the efficiency of the tool use, which was quite cool to demonstrate. So the next question was, okay, this was at the colony level, but then, had the impression that not all the ants were doing the job at the same level. So ask myself whether there is this inter-individual variability in cognitive performance that we can observe in many other animals, and if there is a link between what is called animal personality and cognition. Why we should expect this link? Because what is personality? Personality is defined as consistent behavioral variation. And this is connected to how the individuals explore, interact, and respond to the environment. And we expect a link between personality and cognition because cognition is the way individuals acquire, process, and act on the information from the environment. So it's kind of logical that these two things are connected, but needs to be demonstrated and the phenomenon understood. So we can ask some kind of question like, is cognitive functioning, so the sensory acquisition, the information processing, the learning ability linked to personality? And we started doing this with this tool use behavior, but now we're looking at the individual level and not at colony level. So who is performing this tool use? Are all the foragers equally involved? And is there a link between this individual personality of the different foragers and the tool use in the anti-fungus studies? So to answer this question, we first had to measure at the individual level the personality trait. So, for example, we used very common personality trait like exploratory activity in this sort of open field. So the time that the ant is spending walking or staying in this central area rather than the periphery. So the same, we have this acclimatization tube for about 1 minute, then we remove it and we record the trajectory of the ants. And the other personality trait we looked at was the reaction to prey. Again, here there is a Drosophila, so we're going to look at how much this particular ant is interested in this prey item in this kind of new environment. And to be able to talk about personality, this trait have to be measured at least 2 times, and we have to show that for each individual, I mean, at the population level, of course, this is repeatable, like you see in this graph here. So, for example, if we test the exploratory activity in the open field twice, for example, on day 1 and after 3 or 4 days, then you see that this is highly repeatable. Okay, so we have established that these personality traits are repeatable. Now we're going to look at the behavior during tool use. Sorry. So to do this, we had groups that were formed by 20 workers and some larvae. And these workers were individually marked and characterized each of them for personality traits that I just showed you. And now we had a sequence of tool use trials over 7 days, 2 per day. So here, for example, we have a tool use trial, and now we look at which ants are transporting the tools to the bait. And then we remove those ants. So the tool users are removed. You have to know that normally there's not many tool users at each trial. When we have here 10 pieces of sponges, well, there is normally 1 ant that does all the job, or at maximum there are 2 of them. So each time we would remove. And so the question is, who is that is going to perform tool use in the next trial then, because the tool use has been removed, and so on, until the last day, in which we put everybody back to the small subcolony, and we did this last trial with all the workers were present. So by doing this, we could demonstrate that this personality score that we calculated based on the exploratory activity and the reaction to prey significantly predicted the probability of using tools. So basically, the higher was this personality score, the higher was the probability of using tools. So when we remove an ant that are the first tool user, then in the next trial, the one that is going to replace this one is the one that has the kind of higher personality score. And we can even divide based on the personality score the non-tool users and the tool users into significantly different groups. So this demonstrates that workers with a positive personality score, so they are, what does it mean positive? That the highest explorers and proactive represent to the prey are more likely to be involved in tool use. And so that indeed, I mean, these are all foragers, so there is kind of difference in the group of foragers. So there are some that, are more, prone to do some kind of behaviors compared to others. But now remember that at the end, we put all the ants together and did another trial again. And in this last trial, almost 70% of the tool users were workers that previously performed tool use and that have been removed. So some of these workers were active tool users during the first trial that was 7 days earlier. So this means that it's not something that is, temporary, but they just, this propensity to perform tool use is conserved over time. And this is different from other tasks in other answer, where these removal experiments have been done, and when the answer were returned, they were not doing the behavior anymore. So it looks like the tool users, they really do, they are specialized to do that. So this suggests that there is a fine-tuned division of labor within behavioral groups. So that's not all the foragers, they don't do all the same. And that there is an interplay between personality and division of labor that needs to be studied more in details. Okay, so the next point was about the link between personality and learning performance. So we know that ants are very good learners at a colony and at the individual level. And a technique that is particularly interesting to study in learning is the maxillar-labial extension response with our nested ants that was developed more than 10 years ago by Fernando Guerrieri when he was postdoc in my lab. And this is the equivalent of the proboscis extension reflex in the honeybees. So basically, we present an odor, like a conditional stimulus and a neutral stimulus in the beginning, and associate this with a sucrose reward, and then the ant is standing the maxillar labium to kind of drink this reward. But after a few training trials, the ant is standing the maxillar labium, so the equivalent of the proboscis, just after the presence of the odor that is predicting the arrival of the reward. And so we could use this to study, for example, differential conditioning in Camponotus aetiops foragers. What is this differential conditioning? Is like the absolute one, but here there is a conditional stimulus that is rewarded and another one that is not rewarded. So you have this very typical kind of curves. So in the beginning, the two stimuli, they don't have a value for the ant, but then, and like if, for example, there are two different odors, and here there are 6 training trials, and then the CS plus, this other one is associated with sucrose, and the other one is not associated, or is associated with anything, or with a punishment like salt or quinine solution. And so the ant learn to respond more and more to the CS plus, but not to the CS minus with the extension of the maxillar labium. And so here you see the response at the population level. So for example, for the second trial, there's 20% of the ants that respond. For the third trial, almost 60% and so on. But then you can look at each individual ant and calculate this acquisition score, so the sum of the conditional response to the CS. And you get this kind of representation. And then when I've seen this, wow, these are all foragers, and wow, there is a real big variation. Some of them, they just don't learn, and some others, they learn very well. And all the thing in the beginning, in the middle. So is this variation linked to personality trait? And we studied that again with using the open field for exploratory activity, which was demonstrated to be repeatable. And so we tested individual ants in this open field, and then the same ants, they were tested for differential conditioning of the maxillar labial extension response. And so we can calculate the individual learning score, and we can put this learning score in correlation with the exploratory activity. And so we see here that yes, there is a link between the personality and learning performance, but it looks like strange because, wow, it looks like the ants that are more exploratory, they learn worst. And actually is the low exploring ants that are learning better. At first I was a bit surprised, but when we think about it, we can realize that differential conditioning involves things like cue perception and information storage. Has to be very attentive to do this differential conditioning well. And so in nature, these ants that explore less, they have more time to sample the local environment, are more efficient in forming these associations, for example, between a plant odor and nectar reward, while the high explorer ants that go far away, they go fast, and so they're probably better at discovering novel food sources, but not really at learning these associations. So when we think twice, this is not that surprising. So next, we wanted to know whether there is a link between personality and cognitive bias. Now, what is this cognitive bias? So cognitive bias is the propensity to anticipate either positive or negative consequences in response to an information that is ambiguous. So basically, is this glass half empty or half full? To study this in animals, usually we use a go, no-go procedure. So the animals are trained to discriminate between two different stimuli. And then we put stimulus in an ambiguous position, for example, and the animals are tested in this situation. So if they go, it means that they have a positive cognitive bias. And so they are so-called optimistic because they go, it's like something ambiguous, but well, why not? I go. And if they don't go, or if they take long time to go, then they have a negative judgment bias. And so they are supposed to be pessimistic. I don't go use their energy when I think that indeed is probably the negative thing. So to do that with ants and see whether there was a link with personality, what we did is again, a bit boring all this open field, but we now know that it works and that it's relatively easy to characterize the personality trait of exploratory activity. So we do this first, and then we test the ants in this kind of situation. So this is actually the training phase. For example, here we have on the right, there is a little landmark that it is the same on the right and on the left. For example, for this ant, on the right, there is the sucrose solution, so the reward. And on the left, it is the quinine solution. So they go over several trials and to learn that in this position, there is the reward, and in this position, it's not worth going because there is nothing that is good to eat. And so, of course, for another ant, it was inversed. So the reward was on the left and the punishment was on the right. Okay, but let's see, for this ant, she has learned that on the right there is S plus, on the left there is S minus, and then for the test, there is this landmark, which was exactly the same on the right and on the left, in the middle. So in a very ambiguous position. So is this yes plus or is this yes minus? There is no reward here, there's just a drop of water. And this is the cognitive bias test. So now we just observe and we note how long does it take for the ant to go to check what is if there is the reward here or not. So the very optimistic ones, they will go very quickly, and the pessimistic ones, they will not go, or they will go after a long time. What we have as results, first, we observe that there is a bimodal distribution. There are some ants that go very quickly and some others that go very, very slowly. So two kind of different. And then what we wanted to know if there is a kind of connection between the personality and this cognitive bias. And so when we look at the data, we see that, when considered the exploratory activity, so high exploratory, they are very slow, so negative cognitive bias, pessimistic ants, and the low explorers are very fast. So positive cognitive bias and optimistic ants. And then it's a little bit on the same vein of what we have seen before with the learning. So the high explorers, they have a larger foraging area, they spend more energy during their trips, and so that, as we have seen, a less accurate examination of the environment. So they retain only information about high quality food. They don't risk to go towards kind of ambiguous information stimuli. Like the low explorers, they have a small foraging area, they spend less energy, so they might be more accurate in evaluating the food sources, and so they can retain information about food sources of varying quality, and they can take the risk. So it is basically what is called the speed accuracy trade-off. So this was really amazing to see. And so in summary, what we can say. So the ants do show complex cognitive ability at the individual level, including quantitative cognition, and they are not less intelligent than bees. So the problem are not the ants, the problem are the ant researchers that have improved the way they ask questions to the ants. We have seen that high explorer workers are more involved in tool use, that there is a link between inter-individual variability, so the personality and cognitive performance, that slow explorers learn better than high explorers, and we've seen possibly why. That slow explorers are more optimistic than high explorers, and we have also seen possibly why. So these different individual strategies result in a mixed strategy at the colony level. And so there might be advantages when there is heterogeneous distributions of resources. So in the colonies needs both slow explorers and high explorers because they have different strategies. Well, what is next? So I'd like to study more into details this division of labor, plasticity and personality and how these things interact together. I would like to study more in detail what is the link between personality, behavior, plasticity, and cognitive style. I'd like to also study something I didn't talk about, the interplay between individual and group personalities. So we are individuals that behave in a certain way, but when you are in a group, do you behave different or do you behave the same? So we have done a little study on this on formiga scans, showed that indeed the individual behavioral type is reflected at the group level when they do one task, this brood retrieval. But more recently, and we're writing now this manuscript, in Hormigas, we could show that the groups of high exploratory workers are more aggressive toward the intruders, are more efficient in collecting prey, are faster in relocating to a new nest, more likely to perform tool use compared to groups that are composed of low exploratory workers. So we have to clarify why are there low exploratory workers, what is the importance of this work that they surely are useful for something in a colony. And so this is what we are doing now and what we will do in the future. And also another question I have is try to understand whether cognitive abilities are domain general or domain specific in ants, and how are they modulated by personality. So what is the domain general, domain specific is like, you know, the factor G, the general intelligence that has been described for humans. And so the question for ants would be, are there exceptionally intelligent individuals and ants that can do any task, like elite workers, or individuals with exceptional skills in a particular area? So ants that are very good at doing this task, but not another task. This is something that's interesting to me. And so before concluding this talk, I would like also to tell you that you might have heard about it. I didn't really have time to talk about this now today, but one of my former PhD students, Baptiste Pécret, has been studying the olfactory detection of human cancer by ants, and indeed using and exploiting the incredible learning ability of ants. It could show that ants can detect compounds from cultured human cancer cells. And so these ants are trained with the olfactory associative learning. It can discriminate a cancerous cell line from a healthy one, and also two different cancerous cell lines. And more recently, he also showed that they can make this different using a stimuli in the urine of healthy mice, of tumor-bringing mice. So hopefully in the future, ants will be able to save lives. And with this, I thank you very much, and I thank you all my students and collaborators.
Well, thank you very much, Professor Patrizia, for this amazing talk.
Thank you.
And, you know, so
I kind of don't hear anything anymore, but maybe I close this.
Can you hear me?
Now, yes. What did you say?
Okay, no, well,
Yeah, I don't hear anymore. I don't know. Maybe not my connection, because it looks like it's good. I don't know. Is
Hola?
that we? Yes.
Can you hear me?
Yes, now yes, but when you start talking, it's
Okay, I'm going to repeat. Well, can you hear me well?
Yeah.
Okay, no, that I was.
No, maybe you can write in the chat.
Entra tú.
Sorry, Diana, I couldn't hear you. But I see there is something in the chat. You want me to read that? Can the people hear me then?
Can you hear me?
Yeah, when you say this, yes, but when you start talking, it doesn't work. Try.
Okay.
No, you see, it goes. No, I only. Yeah, I'm sorry. When you start talking, I don't hear you. I see what Shima's written there. Do you think there are species at the species level, not the individual world, there are higher investment on high explorers, others on low explorers, or policy species should need one kind of explorers that the economical? Yeah, sure. But this is logical. So there might be this proportion between high explorers and low explorers might be linked to the ecological lifestyle and environment. You are more a specialist than me on that. But it would be interesting to kind of identify these different species, and then also to look at the individual level what the workers can do in these different species. They are supposed to have different proportion of these workers. Shall I go on? Yeah, no, I don't hear you. I'm so sorry.
Sabrina, can you hear me?
No, yes, but it starts always with yes, and then.
Okay, we have another question from Sabrina. She asked,
Yeah, I see.
Is there also a difference in the
Is there also a difference in the types of items that non-tool users versus tool users to look for while foraging in nature? Yeah, it's a very nice question. I don't know. I guess that you are right, and that in nature, the non-tool users would be more prone to maybe forage for kind of food that is not liquid. Yeah, that would make a lot of sense. Actually, this is something that one can study also in the laboratory. Yeah, that's giving me a nice idea. Thank you.
I have a question. The tool, can you hear me?
I don't know why it starts that I can hear you, and then maybe you can just write it in the chat.
Okay.
Yeah. Yeah, so this is not, there might be something with the microphone of Diana that is going on. That is probably, yeah. Because it's also when she starts and then, no, we don't hear you.
I'm sorry.
It's not your fault. Have you tested to use in other tasks? I'm not sure I understand the question, sorry, Diana. I mean, the thing is that the tool use is true, is flexible about the material that they're going to use and to choose, but they only do this kind of behavior. So they take the material, they put it into the liquid food, and then they take this soaked material and bring it back to the nest. So I have tested tool use in other ants in other tasks, but not the Hormigas. They don't. I mean, I don't think they do other things than what has been described. But maybe I don't know. Yeah.
Okay.
Okay, thanks. I'm going to take over Diana because her microphone is messing up. So I'm Giampiero. Thank you for that amazing talk. We have another question from YouTube. It says, how do ants communicate to their companions in the
foraging part? Yeah, so it depends. The easiest way to communicate is to lay foraging trail. So if there's the food of high quality, then coming back, for example, very common Lazarus ants do this, coming back to the nest, they drop little pheromones, dots, drops to mark this. So in this way, they can communicate the way to go to the foraging site. But then there are also other types of recruitments. So it could be that there is a leader that is taking a little group of ants. So group recruitment, this leader is bringing the ants to the food. Or there is also something that is known as tandem running. So where there are just two ants, for example, the Temnotaurus ants, they do this. So there is one that knows where the food is, that is bringing the other one. So the followers is following and tapping with the antenna on the abdomen of the leader, and they go together. They do this also when they go to a new nest. So there are many different kind of communications, and that depends on the ant species and the ecology of the ants. Okay. Hello.
Yeah, sorry, my apologies. I'm just looking to see if there are any questions on the YouTube site.
Okay. So
yeah,
there is YouTube as well.
Yeah, we haven't received any more questions. So let's just give a couple of minutes more to see if anyone has any more questions.
Is there any difference in age between tool users and non-tool users? No, we didn't measure that. It's a very good question, but they are all foragers. So foragers are supposed to be the oldest ant in the colony because they start doing things inside the nest and then they go outside. But it is true that we don't know the exact age of the workers. So it would be very interesting to try to follow the ants along their life to know how do they change the task, and maybe this is also a matter of age, but they for sure they're all foragers, but we don't know the age.
Okay, thank you. And a question from Diana actually is, so you've studied exploration, and she wants to know if there's some sort of syndrome behavior associated to the exploration.
I mean, is she referring at behavioral syndrome? So behavioral syndrome is an association between different personality traits. So, for example, you expect that the ones that are more explorative, and they are also the ones that are more aggressive, and so on. So we had some of these associations, but we didn't study that in detail yet. Yeah.
Okay, thank you. Let's wait until 45. If we don't receive any more questions, we'll move on to a short 5-minute break, and we'll be back at 9:50 so we can begin our next session.
Thank you very much. It was lovely to be with you, and congratulations for the organization.
Thank
Bye.
you everyone for being here.
Bye. Bye, Jim.
Bye.